Hello, everybody, and welcome to Creation Myths. I am so excited to be here with all of you tonight because we are reviewing the new book Traced by Dr. Nathaniel Jeanson from Answers in Genesis. So, before we go any further, just let me know in the chat if you can hear me okay or if there's any audio issues. And as always, if there's any tech issues during the show, just let me know in the chat because I do keep an eye on it during the show to make sure everything is cool. So like I said, tonight we are going to talk Traced by Dr. Nathaniel Jeanson, and we have a lot to do tonight. So I'm going to start with an introduction, just like who's Nathaniel Jeanson, where does Traced fit in with his work, and then I'm going to just kind of briefly go through the case that he lays out in this book, just broad strokes. Then we'll go through kind of little bit by little bit, and we'll examine the case he makes and how he makes it, and see kind of how the evidence he presents stacks up. And then we'll look at some other kind of issues that jump out at me when I read this book. There were a couple things that were kind of jumped out, even if they weren't the big point of the book. There were some smaller things that I think are still important that I want to mention. And that'll be it. That's what we're going to do today. This will be I don't know how long this is going to take. We're just going to go through it and we're going to see. So let's begin with just a look at this book and its author. So this is Dr. Nathaniel Jeanson. He earned his PhD in cell and developmental biology from Harvard in 2009. He then went to work for ICR, that's the Institute for Creation Research, and then he hopped over to Answers in Genesis. And by the way, everybody, if I have to stop to cough or sneeze or anything, I apologize. I'm on the tail end of a cold, so I'm sorry, but I hope I don't have to. I have tea, so hopefully we're good. But if I do, I apologize. So we've got Dr. Jeanson. He's working for Answers in Genesis. In 2017, he released a book called Replacing Darwin, which I have previously reviewed in 2 parts on this channel. The reception to Replacing Darwin was generally not positive, not just from like the evolutionary biology community, but also from other young Earth creationists. They generally didn't kind of jump on board with the mechanism and the model that Jeanson proposed in Replacing Darwin. And then now it's 2022, and we have his second book, Trace. So I should mention, in between, he published a kind of condensed version of Replacing Darwin called Replacing Darwin Made Simple, which is designed for kind of a less technical audience. I reviewed that also on this channel not that long ago. So we've got Replacing Darwin Made Simple, and now Traced. And Traced just came out. And so let's talk about Traced. Actually, before we do, let me just tell you that a lot of Jeanson's work revolves around a lot of the ideas that he talks about in Traced, rather than the stuff that he covers in Replacing Darwin. A lot of his writings for Answers in Genesis are about things like mutation rates and what's called the time to most recent common ancestor. So you have a bunch of sequences, and how far back in the past do they converge at a common ancestor. That's a lot of what his writing is about, and we find a lot of that, as we'll see, in Traced. So let's talk about Traced. The full title of this book is Traced: Human DNA's Big Surprise. And basically what Jeanson does is he uses phylogenetics and genomics, so genome sequencing, to trace, see what we did there? Trace the history of humanity through what are called Y chromosome haplotypes. Now, a haplotype is just a set of genetic traits that are found together, right? You could think of them as mutations. A more technical term might be polymorphisms, but basically you have a bunch of mutations in the Y chromosome that within a population are all found together, and you can call that a haplotype. And we can sequence the Y chromosomes in all the Y chromosome carrying people in Homo sapiens, and we can then reconstruct the evolutionary history of the Y chromosomes using these lineages or these groups called haplotypes. So what Jeanson does is he takes the data that we have on Y chromosome haplotypes, and he claims that all this data together supports a young Earth timeline and history for humanity. And when I say young Earth, I specifically mean the young Earth model favored by answers in Genesis. That's one of many young Earth models out there, but specifically we're talking about the AIG version of young Earth creationism here. Now, this book has been extremely hyped by AIG, at least. From Ken Ham, you can see down here, Ken Ham, this is from January of this year. He said, "It's the Rosetta Stone of human history, discovered by a creationist." And Jeanson himself on AIG wrote this, talking about how creationists are actually doing a lot of real science these days. He says, "Creationists are now making major scientific discoveries. My new book, Traced: Human DNA's Big Surprise, describes one of the biggest and newest of these discoveries, one that is adding not just new chapters, but new volumes to how we understand our past." So we're not exactly being modest here in how we're promoting this book, right? They're being extremely hyperbolic, right, about the degree to which this book upends our current understanding of human history and provides robust evidence for the AIG narrative. So that's kind of the background before we get into like what the book actually says. So now we can get into the review. Now that we've established what the kind of where this sits in the pantheon at this point. So we'll start with the big picture. Does this book deliver on the promise? Does Jeanson accomplish what he claims? Right? Does he show that the Y chromosome supports AIG's narrative? Nope. No, he doesn't. And many of you watching probably figured that was the case, but we're going to talk about why and how he doesn't in a lot of detail. Before we do that, we are going to lay out the case that Jeanson makes. So I'm going to lay out the framework here, and it's kind of a bing, 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 bang, and there's a logical flow to it. And then what we're going to do is we're going to look at each of these points and see kind of how Jeanson forms the argument, right? So the first thing he does is he breaks out the mutation rate argument, which is a very common thing that Jeanson does. He says you look at the per generation Y chromosome mutation rate, that indicates a most recent common ancestor, that's MRCA, most recent common ancestor, less than 6,000 years ago. That K, that's like 1,000, right? So it's KYA is 1,000 years ago. So specifically like 4,500 years ago is where he puts the Y chromosome most recent common ancestor, because according to the narrative that he's trying to support, it has to be Noah. And the flood was about 4,500 years ago. So that checks out. So he's saying that's the first part of his case, that the mutation rates support that young Earth timeline. He then takes those faster mutation rates and reinterprets the entire Y chromosome chronology based on those rates. So when you have a new haplogroup, forming, when an ancient lineage splits and it diverges into two new groups, conventional evolutionary biology and population genetics assign a date for that node, right? That branch point in the Y chromosome history. Well, Jeanson's going to reassign all those nodes, right? And they're going to be much earlier than they're conventionally dated because he's basing it on this faster mutation rate. So we're not only reassigning the date for the most recent common ancestor, we're also then shifting the entire history up and reevaluating the dates for all the other kind of divergence points, the branch points on this phylogeny. The third thing he does is he then correlates historical events in human history, migrations, invasions, colonizations of islands, for example, right? Historical events that we know happened. And he's correlating these events with the nodes, the branch point on the Y chromosome phylogeny. So he'll be able to say, okay, we have this branch point occurring, and that is where this new haplotype appeared. Oh, and that correlates with this group, migrating into Europe, for example, right? So he's correlating the phylogenetic tree that he's now changed the dates on with actual events in human history, right? This is where the rewriting the history part of his narrative comes into play. Then what he does is he takes the Y chromosome phylogeny, which the Y chromosome phylogeny is what it is. He changes the dates, but the branching pattern is what it is. But he takes that phylogeny and he overlays it onto the Genesis-derived pedigree for Noah and Noah's sons and grandsons and on and on and on. So he says, we have this phylogenetic tree. Well, we could break it into these three groups. Each of the three groups at the node, that's going to be one of Noah's sons. And then the descendants within each of these three major groups are going to be Noah's grandsons and subsequent descendants, right? So he's correlating not just human history, but biblical pedigrees and the biblical histories with the Y chromosome genetics. That's that next step in the argument. Then what he does, the next part is he does what he did for historical, human events that we know happened, right? Migrations and such. He does that same thing. He does number 3, but he does it for biblical events. So we're talking about the flood. We're talking about Tower of Babel and the subsequent dispersal, talking about the Exodus, right? He's taking biblical events and then picking out those events on the Y chromosome phylogeny, which again, he's matched up to the Genesis pedigree, the family tree, starting with Noah. And then finally, once you do those things, you can claim you rewrote the history of humanity and confirmed the AIG interpretation of Genesis. So that's the short version of how Jeanson makes his case. So now we're going to go through this in quite a bit more detail and examine kind of how he does it and see if his methodology and his techniques actually, make sense and allow him to make the claims that he makes. There's one big problem before we're going to go through 1, 2, 3, 4, 5. Before we do that, there's one giant problem that I want to point out. It's a very simple error, and it really does kind of set the tone for the entire book, and it underlies a lot of what's wrong with the book. And the error is this. He conflates genealogy with phylogeny. And these are two different things, and you can't just use the terms interchangeably. Genealogy, which we could see here, we have 2 generations of butterflies right here. And by the way, this figure, this is from Zimmer and Emlyn, 2016. It's a textbook called Making Sense, Evolution, Making Sense of Life. It's an excellent intro to evolution textbook. Encourage you to get it. You can get used versions very inexpensively. So that's Zimmer and Emlyn, Evolution, Making Sense of Life. This figure, everything on this slide, I've chopped it up, but it comes from there, and I really like it. So here we have a representation of genealogy. We're using butterflies for this, right? You have one generation, you have the next generation, right? You've got parents and offspring. A genealogy is dealing with individuals, right? It tells the story of a family, right? You've got parents and offspring and maybe grandchildren, right? That's, think about a pedigree, right? A family tree. That's genealogy. It's the story of individuals and how individuals are related to each other. Phylogeny is the evolutionary history of groups. They could be species, subspecies, populations. They could be big groups, domains, kingdoms, phyla. They could be in the middle somewhere, but it's about groups. So when you look at a phylogeny, unlike a genealogy where you're looking at individuals, over here in this phylogeny, each of these lines represents a population of individuals. So this line represents a lot of butterflies, right? And this branch point right here, that's called a node. So the term node just means branch point in phylogenetics. That's where 2 populations diverge from each other. It's not 2 individuals diverging, and it's not one individual having 2 children. It's a group of organisms that splits into 2 groups of organisms that are no longer interbreeding with each other. Okay, so that's what we're representing with a phylogeny. You're representing the evolutionary history of groups of organisms. Now, that's not to say that these 2 things are unrelated. They're absolutely related to each other. So you take your genealogy, right? You look at just a couple generations of butterflies. Well, let's zoom out. Let's look at more generations of butterflies, right? So we looked at just these few right here, but there's a lot more in here. We could zoom out again and look at a whole population of butterflies over many generations. We could zoom out of that population of butterflies, and we can look at the entire species of this particular type of butterfly, right? So this is many populations that are going to be, interacting with each other, interbreeding, gene flow, whatever, right? But they're maybe geographically distinct from each other, at least some of the time. And if we zoom out even further, we can look at the history of how different species of these types of butterflies relate to each other historically. And if you look at this phylogeny right here, represented by lots of individual familial relationships among those butterflies, we'll look at this phylogeny up here. This is just a graphical representation of what these populations, these species, have been doing over their evolutionary history. So as you can see, genealogy and phylogeny are related. Genealogy is the minute-by-minute, year-by-year history of individuals, whereas phylogeny is the long-term evolutionary history of groups of individuals. They are related, but they are not interchangeable. Jeanson treats them throughout this book as interchangeable. And everything wrong with his argument flows from this error. This is the first biggest error that he commits. There's a bunch of other stuff I'm going to talk about. This is the big problem, and I'm going to show you where this comes up as we go through his points one by one. So now let's break down his case in detail, starting with the Y chromosome mutation rates, indicating your most recent common ancestor. Now, just a note going forward here, I'm going to show you a bunch of pages of the book, right? I'm going to show you a bunch of pages. These are just screenshots from my Kindle app as I read the book. Don't read the entire pages here, okay? Some of you are reading. Don't read. The stuff that I'm going to point out to you is going to be either underlined or boxed in red. I'm going to read that. You don't need to read anything else, and even the yellow highlighting, you can ignore. That stuff that I highlighted for me as I was reading, some of it's relevant for tonight, some of it isn't, so don't worry about it. So what I am going to do now is I'm just going to read you the stuff in red and give you the context, and then we'll talk about it from there. So again, once when I show you the pages, don't read the whole pages. Just that's going to be a waste of time. I'm going to move on before you can finish the whole page anyway. So what he's talking about in this section of the book, and you can see down here, it's probably really small for you, but this is page 74 on my Kindle app. I don't know what that corresponds to in the hardcover, but that's where we are here. It's in chapter 6. And he's talking about the findings from 2 specific studies that he claims show this early date, or I should say recent date, for a most recent common ancestor. So we come over here, he says, of this first study, the number of father-son Y chromosome differences was approximately 10-fold higher than the expected number, considering the range of published Y chromosome copying mistake rates. I think the phrase he replaced there was mutation rates, but he said Y chromosome copying mistake, which that's fine. In fact, the date from this study implied that Y chromosome Adam lived just a few thousand years ago. He then describes a second study. He says in 2017, researchers compared the DNA sequences between 50 parent-offspring trios, i.e. they obtained DNA from father, mother, and child. The 2017 Y chromosome copying error rate again implied that Y chromosome Adam existed about 4,500 years ago. So he's got these 2 studies, and they looked at parent-offspring. And because we're dealing with Y chromosome, it's father-son pairs. He's looking at the number of mutations that occur in this one generation, right? So this phrase, copy error rate, I want to point this out right here. Copying error rate, that phrase is what's called the mutation rate. That's the rate at which mutations occur per unit time. It could be per replication, could be per year, could be per generation, whatever it is, it's the mutation rate, the rate at which mutations occur. And he's saying it's mutations per generation. And that's fine. You could do per generation. No problem there. The problem is that this is different from what's called the substitution rate. The substitution rate is the rate of fixation. Another way to say that, it's the rate at which mutations accumulate within a population. So fixation means that when a mutation occurs, it then increases in frequency until 100% of that site in the genome for the population you're talking about has that mutation. When a mutation's frequency is 100% within a population, that mutation has achieved fixation. Now, why does this distinction matter for what we're talking about with the Y chromosome, most recent common ancestor, and Jeanson's kind of dating for haplotypes in this book? Well, haplotypes are characterized by fixed mutations. right? Within a haplotype, that's a group of Y chromosomes that all share the exact same set of mutations that have accumulated in that group of individuals over many generations. So what's relevant when you're talking about dating haplotypes and how fast haplotypes arise and how fast they diverge from each other and how fast new haplotypes appear, what you care about is the fixation rate. You care about the substitution rate, not the mutation rate. And the substitution rate is going to be many times slower than the mutation rate because lots of mutations are going to occur in a generation, but many of them are going to be lost via natural selection or genetic drift. So you can't just take this one generation rate of dad to son and extrapolate it backwards in a linear fashion and get your rate of mutation accumulation, because not all those mutations that occur in a single generation are going to stick around in that population. This is an error that Jeanson has made before. This is not new ground for Jeanson, but it is, kind of astounding that here it is right here again, the same error that people, me and others, have been pointing out in Jeanson's work, for years. The thing is, he almost got it right. He was this close to getting it right. It was right there in the text. This is actually the page before what I just showed you. So this is page 73 in again, on my Kindle. Your mileage may vary. He's talking about two different studies that he says are not good enough. They're low quality studies. But check out how he describes these studies that found a different conclusion when it came to the Y chromosome most recent common ancestor. Two Chinese men of a known genealogical relationship going back to the 1800s had their Y chromosome DNA sequences determined. The resultant rate of copying errors was slow. It fit the existence of a Y chromosome atom, the ancestor of all living, it's technically Y chromosomes, but whatever, about 200,000 years ago. In 2015, a study of hundreds of Icelandic men produced the same result. And I'll note here that the consensus date in the literature for the Y chromosome most recent common ancestor, it's like 200 to 300,000 years ago. So this is totally consistent with what we find across lots of other studies that Jeanson didn't choose to highlight. So why is it kind of astonishing that he pointed out these two studies? Well, let's compare father to son, fast rate, right? It's one generation, father, son. You don't have time for selection and drift to be operating in that single generation. Distant ancestry, about 2 centuries, right? So over here, you've got, we know for the two Chinese men, it's going back to the 1800s. Hundreds of Icelandic men, we don't have a time frame on that, but you can imagine the genealogical common ancestry for hundreds of Icelandic men. You can imagine that taking several generations, going back 100, 200, 300 years, whatever the case may be, right? So a few centuries, you get a slow rate. Don't even have to be thousands of years, just a few centuries, you see a slow rate. Why? Because as we just said, most mutations that occur don't achieve fixation. They get lost rather than ultimately occurring at 100%. This is extremely basic evolutionary biology. Like, this is super basic undergraduate level evolutionary biology. What does this mean? The implication of this error is that the foundation of Jeanson's case is flawed. The first thing that he does here is he says all our dates are wrong. The mutation rate shows that everything is squeezed much more recently than we thought. And wouldn't you know it, that recent time frame is compatible with the AIG version of Genesis. Well, if that math is wrong, because he's basing it on the wrong metric, he's simply using the wrong tool for the job, right? A pedigree mutation rate rather than a substitution rate, then everything else he says falls apart. None of the dates he describes, none of the correlations between branch points in the phylogeny and historical migration events, none of that stuff lines up anymore if the date is wrong. So we could stop right here and say, okay, we're done. Jeanson's argument is invalid. We're not going to do that because that wouldn't be fun, but like we could, because that's the degree of severity of the error that he commits right here. And one last thing I want to note before we move on to the next thing is that this occurs in like a 4-page span of chapter 6. I think in my Kindle app, it's like page 73, 74, 75, 76. Like it's all right there. That's where he does the move to reset the timeline. And that's it. That's it. It's the rest of the book is kind of built on just those couple of pages, and it's really fascinating. So let's move on from that problem, and we'll talk about the Y chromosome chronology, because the next thing is that he takes his mutation rates and he says, okay, when did new haplotypes appear and diverge from the ancestral haplotypes? Well, Jeanson obviously completely revises the dates that we've established for these events. He has to, because he uses his fast mutation rate. So he writes, based on these results, 10,000 BC and 3,500 BC must be more recent, at least 2,500 BC or sooner. However, for technical reasons, these dates are also unlikely to be more recent than 1,000 BC. Long story short, the most plausible scenario at present is one in which these dates are bunched up near 2,500 BC. So there's some really interesting things going on here. And first thing I'll point out is 2,500 BC is the approximate date of the flood, right? So we're talking a lot of historical events have to get squeezed in like right there, right after the flood very quickly in order to fit Jeanson's chronology. So what he does here is he takes his incorrect substitution rate, and then from that he derives incorrect dates for Y chromosome history. Basically, he's got this big problem. So we know that Y chromosome history stretches way into the past, 200, 300,000 years, right, from the most recent common ancestor. Jeanson has a problem that doesn't fit the young Earth story. So we have to compress that upper age limit side of the equation down into the last 4,500 years. But as he alludes to right here, the dates are also unlikely to be more recent than 1000 BC. Why is that? Well, he also crashes headlong into recorded history on the other side, right? Because now you're going back to 2000, 2500 BC. There's well-documented history at that point. You're like Egyptians were writing hieroglyphics at that point. Right? So you can't go more recent without running into some really severe problems of having to change the known documented dates of recorded historical events. So Jeanson has a very narrow time frame to work with here where he says, okay, my mutation rate puts an upper limit here, but then like recorded Egyptian history puts a lower limit here. So you only have what is in effect a few hundred years to work with. So that's why he's saying you're bunching up everything near 2500 BC. As we're going to see, in order to make that work, he has to like ignore a whole lot of data. This is a big problem for him, this very narrow window for Y chromosome divergence events. So that brings us to the third step in his argument. We're going to take our revised Y chromosome chronology, right? We're changing the dates of our branch points in our Y chromosome phylogenetic tree, and we're going to correlate those branch points to historical events. Now, before I get to any specifics, and we are going to look at a couple specific examples of what he does here, I want to just point out some stuff that I'm not going to spend more time on, but just like, it's red flags. So he ignores archaeology. He just straight up ignores archaeology when he's assigning his dates. He ignores anthropology, right? There's whole fields of study about how people move and interact with each other and the records they've left behind. We're just kind of ignoring that. He ignores radiometric dating naturally because according to AIG and young Earth creationists, that's not reasonable anyway. And his dates, as a consequence of all of this, putting all this other stuff aside, his dates are based on the incorrect Y chromosome mutation rates that we've been talking about, right? That's where he gets his dates. So with those caveats in mind, let's look at two examples of how Jeanson figures out like the history of different Y chromosome haplotypes. Now he goes through a bunch of different groups. We're not going to talk about all of them. We don't have time. I'm just going to pick out two examples that I think are do a good job illustrating the techniques and the methodology that Jeanson employs. So we're going to start with a group called E1B1 and how Jeanson kind of eyeballs where E1B1 originated. Actually, before I go any further, I should note with Y chromosome haplotypes, the way they're named is starting with a capital letter and then alternating numbers and lowercase letters. So if E1B1 is an ancestral haplotype, the descendants from that are going to be E1B1A. Another branch might be E1B1B, right? So it's alternating numbers and letters starting with a capital letter. So what Jeanson wants to do in this part of the book is he wants to say, okay, where is this E1B1 located geographically? And this is the part of the book where, honestly, it goes off the rails. This is where, like, we really get into just amateur hour. So I'm going to read this, and I'm going to show you Jeanson's analysis. And this is a bit of a long excerpt. I'm sorry. It's worth it. Just, I want you all to have, the specific context. So he says, so he wants to know where E1B1 is because he's looking for E1B1B. And to do that, he's going to compare with E1B1A. And he's going to see, okay, we have these two sister groups, and we're going to try to find the common ancestor. So he says, okay, from where did the ancestral lineage to E1B1A and E1B1B spring? We've just seen evidence that the E1B1A lineage spread out from west/west central African homeland. Conversely, we've also seen the association of the E1B1B lineage with the Arab Muslim expansion, which arose in Arabia. It seems natural to put the early E1B1A and E1B1B peoples in those two regions. So we've got Arabia for one of the groups and West Africa for the other group. The shortest distance between 2 points is a straight line. The line connecting Arabia and West Africa resides mostly in Africa, not Asia. The line also happens to pass through Northeast Africa, the area near Sudan. Color plate 42. Let's look at that figure. Here it is. So we've got our E1B1B homeland that Jeanson identifies here, E1B1A over here in West Africa, and he literally just draws a straight line between them and says, somewhere in the area of Sudan. That's probably where that ancestral E1B1 population came from. He then goes on to say, together, these facts suggest that the early E1B1 peoples, the ancestors to E1B1A and E1B1B lineages, lived in Africa, possibly near Sudan and or Ethiopia. Color plate 44. Here's that figure. So he has E1B1 right here, and then they migrate in either direction to go to the ancestral homelands Jeanson identifies for the descendant A and B groups. Now, you may be looking at this and thinking, I don't think that migration pattern makes a whole lot of sense. And you know what? You're right. Because what does Jeanson have the E1B1 population doing as it diverges into E1B1B? Has them crossing the Red Sea over here. Now, that's a body of water, and we're talking very early in human history. This would have happened conventionally or through Jeanson's timeline, either way. So that's going to be a tricky endeavor. But there is another well-documented travel avenue in this part of the world. It's the Nile River running north to south from Sudan into Egypt. So isn't it more likely that rather than going across and then up and then back into Egypt, isn't it more likely to go up the Nile, or I should say down the Nile, right? It's downstream. You're going downriver, and then across Sinai into Arabia and the Middle East. Wouldn't that make more sense? Yeah, it would. And that's a well-documented trade and migration route throughout human history. So this is for real Jeanson's analysis of how he arrives at this region and then for the E1B1 group, and then the resulting migration path that he proposes. This does not make any sense. This does not make any sense. Now we're going to talk about a group called R1b, and it's present in Europe. And I think it actually gets worse. I think this is actually worse. So again, I'm going to read quite a bit here because I want to give you the context. So, but just background, R1b and the subgroups within the R1b haplotype are present all over Europe, in particular in Western Europe. So what Jeanson does to establish the dating for R1b and its appearance in Europe is just ignore like inconvenient data that refute everything he's saying. That's what he does. So let's look at this. He says in the AD 400s to 700s, the ancestral population of R1b men resided somewhere in Far Eastern Europe or Central Asia. Then in the AD 700s to 1400s, a subgroup of these men started moving westward. Some of the migrating men settled down in places they crossed, like Western Russia, the Caucasus, Turkey, the Balkans, and the former Eastern Bloc countries. He continues. Then in the AD 1400s and 1500s, so like within the last few hundred years, right? We're looking within the last 600 years here, something happened to push another group in the direction of Central and Western Europe. Migration and dispersal appear to have happened rapidly. The sublineages that concentrated in Central Europe, in France, the Iberian Peninsula, in Switzerland, and Italy, and in the British Isles and Ireland all separated within a few decades of each other. And I want to highlight Italy here. Again, the date range here for Italy, 1400s to 1500s. It's going to be important in a minute. So Jeanson's claim here is that this R1b haplotype arrived in Europe between the 7 and 1400s AD, and specifically arrived in Italy in the 14 or 1500s AD, right? So very, very recently, this haplotype arrived in Europe. Now there's a problem for Jeanson, and the problem is the presence of Stone Age remains in Italy from which we have extracted DNA and sequenced it. And wouldn't you know it, those Stone Age remains contain haplotype R1b. So this is from a paper in Nature in 2016, Fu et al., The Genetic History of Ice Age Europe. By the way, any papers and links to like other things that I reference, they're going to be in the description. They're not there yet. I'm going to put them there once I'm done. So this is a paper called The Genetic History of Ice Age Europe. And in this paper, the authors write, we were surprised to find haplogroup R1b in the about 14,000-year-old Villabruna individual from Italy. It's a very famous Stone Age burial skeleton is laid out, and they were able to extract DNA. It's a Villabruna 1, I think, is the specimen. Thank you to the Peaceful Science Forum for bringing this example to my attention. I appreciate you. This has been really interesting to read about. Now, Jeanson, of course, rejects this date, right? He rejects this date of 14,000 years. Right? He says the world is not 14,000 years old. This is silly. This is incorrectly dated. Okay. It's still a Stone Age burial site. It's not a medieval burial site. It's Stone Age. According to Jeanson's own chronology, the Stone Age occurred shortly after the Tower of Babel dispersal. The Tower of Babel dispersal, according to Jeanson, occurred within a few hundred years of the flood. So the flood was about 2500 BCE, and that means the Babel dispersal was somewhere in the neighborhood of 2400 to 2100 BCE, which means that the Stone Age was about 4,000 years ago, about 2000 BCE. Jeanson assigns a date for R1b entering Italy to the 1400s. But we have a specimen that, according to Jeanson's own chronology, would have been there 3,000 years before then. It doesn't work. His chronology doesn't work. He has to just ignore well-documented examples where we know what the haplotypes are and we know the age of the specimen. But he just ignores it because it's inconvenient for the case that he's trying to make. So that's a big problem for the case he makes. This is not the only example of this in this book, but it is an extremely clear-cut one, and the error is so egregious that this is the one I chose to point out. So then we get to part 4, where not part 4. The book isn't in these five or six parts that I've described. It's part 4 of his argument, right? In the flow of his argument. Now, part 4, what he does is he overlays phylogeny and pedigree, genealogy. And he does this quite literally. I'm not exaggerating. I'm not saying he kind of conflates them. He says, well, this branch might be this. But like, he quite explicitly just puts the two on top of each other and says, and makes a one-to-one comparison. So here's how he does it. He just overlays the Y chromosome phylogeny, which you can see right here, and the pedigree of Noah and Noah's male descendants from Genesis right here. And it's color-coded based on the sun. So we've got our three sons, 1, 2, 3, and then the groups that are descended from them, right? So you've got, for example, he says, okay, the Jews came from over here, the Greeks came from over here, on and on. And he has a few figures like this where he has different groups labeled and different candidate groups labeled that he's less confident about, right? So you can see what he's doing here is he's literally just taking these two things, right? The Y chromosome phylogeny and the pedigree from Noah and his sons, and he's just overlaying them in a one-to-one way. You can't do that. These are two different things. These are two different techniques. They're not interchangeable. This is not a pedigree. It's not a history of families. This is not a phylogeny. It's not an evolutionary history of groups. Remember, the nodes, the branch points in here, as I said earlier, the nodes are populations. And when they branch in the phylogeny, it's the nodes diverging, those ancestral populations diverging into two or more descendant populations. Jeanson treats these nodes as individuals. So like right here, right? You've got a node, a branch point. He treats that as one of Noah's sons. Up here, it's a little tough to see, but it's right up there. He treats that node as Shem right there. This node represents a group of individuals. That is a population of individuals, not a single individual. It's completely inappropriate to just treat phylogeny and pedigrees as interchangeable. It's just astonishing to me that anybody would do this kind of directive and association between the two. It's mind-boggling. And then the fifth part that I mentioned, remember I said, and then what he does once he associates the pedigrees with the phylogenies is what he does is he does the historical event thing, but he does it for biblical events. So he has some tables at the end. So he has some tables at the end where he says like different Y chromosome haplogroups. There's a lot of these. I only plucked out 2, but there's like multiple of these. He has like Y chromosome haplotypes and he relates them to the biblical lineage, right? So, Ham, Japheth, Shem, right? He just relates haplotype son, right? He just does the same error, but like very specifically. This lineage is in this part of the family tree. And he correlates these different lineages to specific biblical events, right? So if you look right here, he points down L within Shem might be the Edomites or lost tribes of Israel. Over here, he's got T also within Shem, likely lineage of Abraham, Isaac, Jacob, and Jews, right? So he just takes his overlaid Y chromosome phylogeny and pedigree and then says, oh, these branches represent this biblical group. These branches represent this group. This node represents this event in the Bible, right? That's what he does here. It's based on just absolutely terrible methodology, which I hope is clear at this point. So, ta-da! History explained through Genesis and the Y chromosome. So what we've done here is a step-by-step of how Jeanson's case is supposed to work and a step-by-step of why it doesn't. We're not done. Oh no. We have more to talk about. So there are some more problems with this book. The first one I want to point out is Neanderthals. Neanderthals, very famously, we have DNA sequences from Neanderthals. They are a group of hominins that are very closely related to Homo sapiens. Now, the problem with Neanderthals for young Earth creationists is that we can make a phylogenetic tree of our DNA along with the DNA we've gotten from Neanderthals and other archaic groups like the Denisovans and some others, right? We can make phylogenies that go beyond Homo sapiens. When we do that, we find that Homo sapiens all group together, and then these other groups are highly divergent from Homo sapiens, which means that the most recent common ancestor of Homo sapiens and these other groups must be in the more distant past compared to the common ancestor for just Homo sapiens. Okay, so look right here. We've got mitochondrial DNA, Y chromosome DNA, and nuclear DNA, except for Y chromosome. So this is autosomes. And this is all from a paper 2018. Again, I'll link the paper down below so you can check this out yourself if you want. What we see in the gray in each of these phylogenies here, here, and here, that's modern humans, right? That's Homo sapiens. And this triangle represents all of modern humanity. So the point of the triangle on the left there, that's the most recent common ancestor for all of humanity, here, here, and here. Now let's just focus on this tree right here, because this is the Y chromosome tree. And this book is all about Y chromosomes. Just whatever I say about the Y chromosome, it also applies to the other two. But let's focus on the Y chromosome. Here we have our Neanderthal Y chromosome samples. This node back here in the more distant past, so this is the present, this is the past, this node back here represents the common ancestor of the Neanderthal Y chromosomes and the Homo sapiens Y chromosomes. It's in the more distant past than the common ancestor of all the Homo sapiens. So Jeanson used these sequences to get a date of 4,500 years, which means if you consider the Neanderthals, you get a most recent common ancestor that is too far in the past to be compatible with the young Earth timeline. The problem here for creationists is Jeanson and most young Earth creationists, not all, but most young Earth creationists agree that Neanderthals are descended from Adam and Eve. The reason for this is that we interbred with Neanderthals. Homo sapiens interbred with Neanderthals. Most humans have some Neanderthal DNA sequences in their genomes. We have markers in our DNA that are characteristic of Neanderthals. How did they get there? Through interbreeding with Neanderthals as Homo sapiens migrated throughout the world and encountered not just Neanderthals, but other archaic members of genus Homo, like the Denisovans. That's down here in yellow are the Denisovans, right? So we interbred with them, therefore they have to be part of the humankind, according to most young Earth creationists, which means they're descended from Adam and Eve, which means they need to converge at our most recent common ancestor. But they are so divergent that they push the most recent common ancestor too far in the past to be compatible with the young Earth timeline, even if you use Jeanson's incorrect mutation rates. So like even by Jeanson's wrong math that we've talked about, Neanderthals still break his model. So I'm going to show you on this slide every mention of Neanderthals in this book. The word Neanderthal appears in the text of this book on 2 pages. And there are 3 total uses of the word. The first, well, so let me just answer the question, how does Jeanson address this problem? In his 3 mentions of Neanderthals, he doesn't. I'll show you. I'll show you every mention. Here's the first part where he's describing the mainstream model of human origins. Before anatomically modern humans arose, primitive Neanderthals had evolved from even simpler life forms. Before Neanderthals, Homo erectus had evolved from an ape-like ancestor. This ancestor came from an even more primitive predecessor, one that also fathered the genealogical lineage leading to modern chimpanzees. So we've got 2 mentions, 2 out of the 3 right there in that sentence. He's just describing, his version of mainstream human evolution, which I take a couple issues with that paragraph, but it doesn't matter. Here is the 3rd and final mention of Neanderthals in this book. He's talking, the lead in here is he's talking about early, the dating of early civilizations. So Mycenaean Greece, Egypt, Mesopotamia, Indus River. That's when he's talking about cradles. That's what he's talking about here. He says, presumably these cradles arose shortly after Babel. This would have placed them within a few centuries after the beginning of human history. Therefore, events dated even earlier in the mainstream version of history, such as the rise and fall of the Neanderthals, the Stone Age, and the Agricultural Revolution, all would have transpired within these early centuries. So he's putting the Neanderthals very clearly after the flood. So within the last 4,500 years, which means the Neanderthal Y chromosomes must share a most recent common ancestor with modern humans, which means this can't be dated to 4,500 years ago. This has to be dated to 4,500 years ago. But Jeanson dates this to 4,500 years ago. He ignores the date for this, which breaks his model. There's nothing else to it. He doesn't say anything else about Neanderthals at any point. That's it. He just ignores the fact that they totally break his case. So I don't have anything else to say either. Let's move on to the next problem. The next problem is that he has no support from other young Earth scientists. Yet, it's early days. The book just came out. Maybe people will, grab onto it, and we'll get, as time goes on, we'll get, more support from other people at AIG. We'll do kind of technical reviews, maybe other young Earth organizations, CMI or ICR, other groups. Hey, maybe they'll do, technical reviews, and they'll check it out, and they'll say, you know what, I agree with what Jeanson is selling here. I agree with his model. But so far, that's not the case. And so far, what I've been able to see is that young Earth creationists typically disagree with it. One I'll point out right away is a young Earth YouTuber who basically specializes in like Y chromosome stuff. I don't know what his technical background is, but it's his YouTube channel is Jungle Jargon. I've had him on this channel before. We've talked about stuff, and he says that like Traced has problems. He's done a number of videos on this in the last couple of weeks. And like his thing, the thing that he does is all about Y chromosome ancestry and which of Noah's sons led to which, extant groups. And he disagrees strenuously with the conclusions that Jeanson reaches. So I think that's interesting. I was actually expecting Jungle Jargon to like be super gung-ho about Trace because it's like it's his thing, but he actually totally disagrees with the model. But that's not the most interesting thing that's popped up. The most interesting thing that's popped up is this piece from Dr. Rob Carter, A Family Tree for All Humanity. Again, this is from Dr. Rob Carter from CMI, that's creation.com. This was published just last week, April 5th. And so I am going to show you a lot of excerpts from this, both from the piece and from the comment section where people asked Carter questions and he provided answers. I'm going to do this because this piece is about a secular piece that Carter references. It's someone wrote in to CMI asking about the secular piece that said that what they did is they basically, kind of similar to what Jeanson did actually, is they made a family tree for all of humanity, and they kind of mapped migration patterns and lineages and that kind of thing. It's a very cool paper. So I'll link this, down in the description. Rob Carter takes this question and goes through it and talks about the methodology and, what's okay and kind of what's questionable. I'm just going to pull out some excerpts that I think are relevant to what we've talked about already. So first, this is just a heading: problems with comparing genealogy and geography. You can't necessarily correlate geography, genealogy, and genetics. This is a long-standing and well-known problem. Yes. Yes, it is. People migrate, and he goes on to talk about why it's a problem. It's true. And I specifically want to highlight this part, genealogy and genetics, because that's literally what Jeanson does in the book, is he correlates genealogy and genetics in a very strong, just overlay them kind of way. So I found that striking, that statement from Rob Carter. He goes on to write, again, don't read all this, just follow me with the red. First, even if, so we're talking here about the authors of this piece identifying the ancestral region for mitochondrial lineages. He says, first, even if the mitochondrial ancestor could be traced to this location, this does not mean the Y chromosome ancestor or the many ancestors that contributed to the autosomes would have lived in the same place. Second, just because people with these mitochondria live in a certain place today does not mean their ancestors lived in this same place in the ancient past. Now, he's talking about mitochondria and how that's not necessarily going to correlate with Y chromosome or autosomes. Well, Jeanson is talking about Y chromosome and essentially doing what Carter is saying you shouldn't do. And the reasons that Carter identifies are valid. Excuse me, I do have to cough. I'm sorry. Trying desperately to get to mute first. We're back. Okay, let's continue with Rob Carter here. The authors themselves in this piece point out some shortcomings with their own work. They say, sorry, I have to cough again. I apologize, everybody. Okay, I think we're good now. Okay, so the authors themselves point out some of the limitations of their own work. They say the accuracy of any ancestral geographic inference method will be limited when the distribution of sampled individuals does not reflect the location of the sample's ancestors. That's correct. Yeah. And migrations occurring within the past few thousand years means that present-day distributions of groups in Africa and elsewhere may not represent those of their ancestors, and thus we may have a distorted picture of ancient geographic distributions. Yeah, that's true. Those critiques, those limitations apply not just to the paper Dr. Carter is talking about, they also apply pretty specifically to trace. Now, this is where it gets fun and notably specific. So I want to point out this map here. This is showing some of the results of a simulation that the authors ran, looking at how groups radiated throughout the world. And I want to just draw your attention to how this migratory path crosses the Red Sea. Okay, so follow me here as we kind of go through this text. In this screenshot from near the end of the simulation, which runs backwards, notice how the plotted points run straight across the Red Sea. This is extremely artificial, as any migration route would be expected to go around the north of the Red Sea via Sinai or the Mediterranean coast, as I said before, or across the 10-mile wide strait between Djibouti and Yemen. That would be down here, right? This distribution of points is an artifact of their method of plotting ancestral locations as the midpoint of a great circle route between two daughter nodes. While interesting from an academic point of view, this is entirely non-historical. Have we seen anybody recently plot ancestral regions as the midpoint between two groups? And coincidentally, maybe cross the Red Sea rather than go around. It's exactly what Jeanson did. This critique from Rob Carter, this extremely specific critique, applies very specifically, not just to the paper Carter is talking about, but to Jeanson's methodology. That example in particular, I find the specificity very notable. Now, Carter does also address a couple questions here, one of which was about ancient DNA, and I'll just read a couple parts here. I do need to clear my throat one more time. I'm so sorry, everybody. This cold, it's lingering. So he writes, not only do I not reject a DNA that's ancient DNA as a reality, so like Neanderthal DNA, for example, I find it is an amazing tool for unraveling ancient history. We can see the progression of people from places like Europe, and he goes on to describe its utility. Ancient DNA should not be rejected by the young Earth community. Instead, we need to incorporate it into our ideas of history to whatever extent possible. Jeanson, remember, just ignores ancient DNA entirely. Neanderthals, Denisovans, invalid, can't use it. And then one more question that Jeanson that Carter addresses, someone asking actually about Jeanson and his strict linear molecular clock. Dr. Carter writes, Dr. Jeanson uses the molecular clock concept as a simplifying assumption in his model. This does not make it wrong per se, but it does qualify his conclusions. He goes on, he explains it in some detail, which I'll link this. You can read it if you want. But he goes on to say what this means is that you can't necessarily look at the length of a branch on the family tree and divide by 4,500 years to get a mutation rate. The problem is that this is basically exactly what Jeanson does. Look at this figure from Jeanson. He literally takes these threes, represent 3 mutations per generation. Jeanson is using a strict linear molecular clock, 3 mutations in the Y chromosome per generation, and extrapolating that to get his 4,500 year time frame. Dr. Carter is saying it's a simplifying assumption, but it does qualify his conclusions. You can't really do it the way that Jeanson does it. And he's right. Dr. Carter is right here. Credit where it's due. So one more note on this Carter piece, which is fascinating to read, is that he's critiquing a secular paper, but the specificity with which his critiques apply to Jeanson's work do make one wonder. I don't know if that's purposeful or not, but you do wonder. So we are wrapping this up, thankfully, because I'm losing my voice. I will be quick as we conclude. Dr. Nathaniel Jeanson attempts to show in Traced that the human Y chromosome history is consistent with the Answers in Genesis view of Genesis. This is an extremely flawed work involving a number of serious errors. He treats the per generation mutation rate as a substitution rate and therefore incorrectly dates the Y chromosome phylogeny. He treats pedigrees and phylogenies as interchangeable, which is an extremely basic error. You can't do that. He ignores established, well-documented dates, histories, events. He just throws out data that are inconsistent with his conclusion, basically. Most egregiously, I think he ignores Neanderthals, which directly contradict his findings. They completely refute his timeline, even though he acknowledges in the text that Neanderthals have to be part of his model. They have to be descended from Adam and Eve, and they have to have existed post-flood. And he has so far failed to garner support from other young Earth scientists. So what does this mean? It means that Traced does not live up to the hype. The very last thing I want to just mention is this question of who is Traced for? Who is this written for? It's clearly not an attempt to convince me, right? This is not an attempt to convince mainstream biology that young Earth creationism is correct. That's not what's going on here. And I don't think it's an attempt to convince non-young Earth creationists, Christians or not, that young Earth creationism is correct. I think very clearly what this is to make young Earth creationists feel good, is to provide reassurance that there's science that backs up the young Earth beliefs. I really think that's what this book is all about. At the end of the day, given the errors in it, I can't come up with a better explanation. There are too many obvious errors. There's too much contradictory evidence that is just ignored to be anything else. So there's one more thing I want to say here, and it's that I did reach out to AIG through their official publicist portal request an interview thing. Jeanson has been doing publicity for this. He has given some interviews over the last few months about this. He generally doesn't make that many appearances. But I did reach out for the official thing, said, hey, let's talk about Traced. So we'll see what happens. If anything comes of that, you will, of course, know, because I will let everyone know, and I will be very excited if anything comes of that. So, thank you all for watching, everybody. This has been my review of Traced Human DNA's Big Surprise by Dr. Nathaniel Jeanson. I see a bunch of people here still watching after an hour of this. I really appreciate that. That really warms my heart that so many people are here watching. If you haven't done so already, please hit the like button. Please share this. If you're not subscribed, do that too. I haven't seen any really in-depth reviews of Traced, and certainly not any from non-Young Earth sources. So if you see anyone talking about it and you want to throw a non-Young Earth source out there, do share this around. I would really appreciate that. That would be really cool. So thank you all. I'm gonna maybe go make some more tea and call it a night. This has again been my review of Traced by Dr. Nathaniel Jeanson. It was really bad. It's full of errors. It is not to be taken seriously. Have a good night, everybody. Thank you for watching.